In this video, we want to show you what the distribution kit is, what it contains, and how you can work with it. Every year, the iGEM headquarters prepare a so-called distribution kit that travels all around the world for each of the iGEM teams. Some of the contents of the distribution kit can vary between each year, but overall you will get the following: DNA plates with biobricks, linearized plasmid backbones, a kit for testing competent cells, a measurement kit, and iGEM stickers and pins. The first two things that you should do when you get your distribution kit are gather your team members and take the most amazing and creative picture and share it on Instagram with the hashtag #WelcomeToIGEM, and pay close attention to the storage instructions of the kit components. Some of them must be frozen, while others must be kept at room temperature. Let's look at the contents in a little bit more detail. DNA plates with biobricks. These should be stored at -20°C. In this 384 well plate, you can find some of the best biobricks from previous years. You can find detailed information on the biobricks that you got in the iGEM website. Some examples of parts that you can get are indecible promoters, enzymes, or fluorescent proteins. For more information on biobricks, check out our video How to iGEM number 3, link in the description. Linearized plasmid backbones. These should be stored at -20°C. In this little box, you will find the biobrick plasmid backbones PSB1C3, PSB1A3, PSB1K3. The letters here stand for the different antibiotic resistances that they carry. C is chloramphenicol, A, ampicillin, and K, kanamycin. It is important to note that the plasmid backbones are PCR products. They must be digested with EcoR1 and PST1 to combine them with a biobrick of your choice, digested with the same restriction enzymes, in order to transform them into your competent cells. Competent Cell Test Kit. These must be stored at -20°C. One of the most useful contents of the distribution kit that you will get is the Competent Cell Test Kit. You can use it to test and calculate the transformation efficiency of any competent cells of your choice. These ready-to-use plasmid backbones, once again the standard PSB1C3, PSB1A3, PSB1K3, come with a very special biobrick in them, the red fluorescence protein, RFP or BBA-J04450. We highly recommend using these plasmid backbones in your project because you can use them as a selection marker for bacteria that took plasmids which were not successfully digested or that unfortunately religated with the RFP and not the biobrick from your experiment. Measurement kit. This should be stored at room temperature. Some of your projects may require fusion proteins which include GFP in them. You can use this kit to calibrate your plate reader, calibrate the OD measurements, and flow cytometry fluorescence calibration. The iGEM website has very detailed tutorials and spreadsheets that you can use for your measurement validation. In addition, the iGEM's measurement committee will help you reviewing the calibration data that you obtained and advise you if there are any problems. How to work with your distribution kit. Let's say you picked a biobrick that you want to use. First, it's important to know that the biobricks come already integrated into a plasmid backbone. For each biobrick, you have to check in the database for which plasmid it comes from and which resistance it has. In the same place, you will also find the location of your biobrick, which plate and which well. When you find the correct plate, you might not be able to read the letters and numbers on the side. Just push down the aluminum foil with your thumb to make them visible. Then mark the desired location. Pipette in 10 microliters of water and let it stand for 5 minutes. There you go, your biobrick is ready for transformation. All links you will find in the video description. We hope this video helped you to navigate the iGEM universe. TU Dresden wishes you good luck with your project.